Hello, in this video we're going to talk about Science Unit 2, Lesson 5, which is DNA, Modeling in the Sciences. And as an overview of this lesson, students will be introduced to transcription and translation. They're going to read and annotate Phelan 5.6 to 5.7, and this is really where it goes in depth onto how transcription and translation work. They'll talk about that using text-dependent questions. They're going to watch a couple of science animations on transcription and translation, and they're going to be introduced to modeling. And so they're going to watch a PBS video on the transcription and translation model, and then they're going to do a protein synthesis activity, and that's really the bulk of Lesson 5, so that's what we're going to go in depth onto today in this video. So what are students going to have to do? They're going to create their own model on the process of protein synthesis based on analyzing and synthesizing the materials they've already read and or viewed. So in your lesson plan is a long activity that explains the whole process, but students aren't going to get all that. They're going to be able to create it based upon what they've known. So they spent a whole long time making a model of the structure of DNA. They've thought a long time about using the structure to think about how DNA functions, and then now they're going to take that knowledge to think about the role of DNA in protein synthesis. They're going to use that model to deepen their understanding of the complex process of protein synthesis and to make predictions. So they're actually using models much the way scientists do. So the creating it's important, but also being able to utilize that model to make predictions when the process is altered in some way is equally important. So they're going to use minimal directions on how to create this model. And it's an effort to allow that productive struggle that's really required when they're thinking about this particularly complex process. So what do you need to know? Well, first of all, the step-by-step directions are for your use only, teacher use only. So the intent is for the students to create the model, not to do a worksheet. So if you just handed out all the directions, that's what it become. It really would become a worksheet problem. That's not what we want. So you have the full activity in your lesson plan. Students have only a modified view that I'll show you in a second in their academic notebook. So you can use the procedure, those step-by-step instructions that you have to guide students as needed, but it's really important to allow them time to grapple, to really think through the process. So the next slide is going to show you the instructions. So you can see that they're fairly minimal. So basically they're going to get a DNA template strand, and then using the materials provided, they're going to create a model of transcription and translation of that gene. And they need to not only create the model, but make sure every part is really labeled. So all of these pieces need to be labeled in their model. Now, if you are not a science teacher, if you're an English teacher doing the science unit, or you teach something else, this would be a great lesson to bring in a science teacher, a biology teacher to help you with it, because really this is a particularly complex process. We've had some English teachers who did this in the field testing who got a little bit of help from a science teacher, but ran the lesson themselves and really felt like everybody had a sense of accomplishment by the end. So you can do it, but I think it's a really good idea to bring in a science teacher just to make sure that you really know what all these ideas are and how it's going to go. So before class, to get this process going, there's some things that you need to set up. And all of these materials are available on the SREB website. So you can go to the website and all the PDFs are there, or you can go to this website listed here, Explore Biology, and download this lab. And so you're going to need the DNA transcription and translation strand. Print those on cardstock because students are really going to need to manipulate them. And you might want to cut them into strips before class, because what we found in the field testing is that actually took quite a bit of time. So you might want to get all that prep before. They also need these the mRNA bases, each one printed on a different colored paper, and then the tRNA printed on different colors of light colored stock because students are going to need to write on those. So all of that you're going to need beforehand just to get set up so that students are ready to go to create their models. Students are going to need time to situate and to work on both the model and the questions in the academic notebook. I recommend that you have them work in groups on this so they can really be thinking through the science. But it's not going to be a lesson where they just jump right in. They're going to need a little bit of time to kind of figure out what they're doing, and that's actually an important part of the process. So, don't feel like you need to jump right in there. Give them that moment to really work it out in their group. Eventually what's going to happen after they create their model is some of the questions are going to ask them to use the model to predict what will happen given a mutation, and they're going to use this information as they read Phelan 5.8 and 5.9 in activity 3, and I'm going to show you an example of that in a minute. But it's not just that they're building this model, but then they're going to need to be able to use it. So the more accurately they can label the model and the better they can be sure that it's accurate, the easier time they'll have when they get to this second part where they're looking at mutations. So here's an example. So they're given this strand and they need to figure out when a mutation happens, what's going to happen to the amino acid sequence that's translated from it. So they're going to need to be able to predict what happens based upon what they have. And they can use those codon charts that they have to help them with that. So those are really just there as a reference, but this is a good point maybe to have them take a look at that. And then for you only, so if you want to find out a little bit more background about protein synthesis, there's a couple of good short videos that you can look at. I like this narrated animation. It's only about 2 minutes long. And then there's a longer one from Bozeman Science. It's about 12 minutes on transcription and translation. Don't show these to students. These are just for you. We want the students to be able to learn this idea, learn these concepts through working through the activity. But these are just some ideas for you in case you want to extend a little bit of your background before you teach this particular lesson. And again, the take-home message in this one is to allow that time for the productive struggle. Jump in if you have to, if students aren't really making any progress, but give them that time to really think it through. At this point, given the material that they've read, given the experiences that they've had with modeling from Unit 1, they really ought to be able to do this. So that is Lesson 5 in a nutshell. Thanks.